Evolving landscape in the management of type 2 diabetes mellitus. Focus on teneligliptin. Today, we will try to discuss the newer launch in India, teneligliptin, under these headings. The structure-activity relationship, that means how all the gliptins binds with the DP4 enzyme. The kinetic and dynamics of the teneligliptin. The trials which are majorly conducted in the Japan, Korea, and one trial in India. We will discuss this trial under the monotherapy combination with other anti-diabetic drugs, Indian trial, and special population of renal failure and hepatic failure. At the last, we will see how the teneligliptin is performing compared to the other gliptins by looking at the data taken from the respective PR, because we don't have the head-to-head trial, which is very rare to have the head-to-head trial between the same class drugs. So to start with, the structure-activity relationship, the gliptins, this is the 5th gliptin launched in India. The 6th one, the allogliptin, will be launched very much shortly. So we will try to classify these 6 gliptins depending upon their binding with the DP4 enzyme. This DP4 enzyme has many subsites. The basic one are the S1 and S2. All gliptins bind with these S1 and S2. So the gliptins which only binds with the S1 and S2 are the vildagliptin and saxagliptin. The class 2 includes the linagliptin and allogliptin, which binds in addition to the S1 prime and S2 prime. The third class includes the sitagliptin and teneligliptin, which in addition to binding to the S1 and S2, they bind also to the S2 extensive subsite. So what is the benefit of binding to this extra subsite or classifying the molecules depending upon their binding? So each molecule will have different IC50 value, that is inhibitory concentration required to inhibit the 50% of the enzyme. The vildagliptin requires the maximum amount of drug, whereas the linagliptin and teneligliptin requires the least amount of drug to inhibit the 50% of the enzyme. In short, vilda will be considered as the least potent, whereas the linagliptin and teneligliptin are the most potent DP4 inhibitor. So this potency will help us to decide the dosing of the molecule. Moving further, teneligliptin has certain specific kinetic properties. These are the long half-life of elimination, almost 21 to 24 hours of plasma half-life. So this is the real once-a-day molecule. If you look at the other gliptins, only linagliptin has the more half-life compared to the teneligliptin. The second property, the food will not have any major impact on the absorption of the drug. So drug can be taken irrespective of the food and any time of day because of its long half-life. The third property, this is one of the rare molecule which has 3 enzymes involved in its metabolism. The enzymes are cytochrome P3A4, the flavin containing monooxygenase 1 and flavin containing monooxygenase 3, FMO1 and FMO3. So drugs like ketoconazole, when used along with teneligliptin, which is a strong cytochrome P3A4 inhibitor, the drug will still get metabolized by other enzymes like FMO1 and FMO3. So there will not be any practical issue of drug-to-drug interaction with the teneligliptin uses. The fourth property of teneligliptin, the kinetic property, it's dual mode of excretion through both hepatic and renal route. That means drug will get eliminated in equal amount through the hepatic and renal route. If you look at the excretion, percentage excretion of all the gliptins, the teneligliptin 45% through the renal route and 46% through the hepatic route. Linagliptin, we all know, majorly limited through the hepatic route, whereas other 4 gliptins, sita, vilda, allo and saxa, they are all majorly eliminated through the renal route. So in short, linagliptin and teneligliptin will not require any dose modification when used in any form of renal failure. We have the special population trials of teneligliptin in renal failure and hepatic failure. We will see in the subsequent slides. In this graph, we will see the effect of teneligliptin on the GLP-1 levels, which are increased almost more than 2 times compared to the placebo group. That means even the drug is taken in the morning, the GLP-1 levels are increased after the breakfast, after the lunch and after the dinners. In short, the effect of teneligliptin will be there for the entire day. Moving further, the effect of teneligliptin on the beta cell function, that is HOMA beta parameter, which is significantly increased after the monotherapy by almost 8 factor, and when it is used in combination with sulfonylurea also, there is a significant improvement of HOMA beta parameter by almost 8.8. And the placebo group, in both the studies there is no significant change in the HOMA beta parameter. Next dynamic property, the insulin resistance, the effect of teneligliptin on the insulin resistance significantly reduced by almost 0.9 factor, and the C-peptide levels, the postprandial C-peptide levels are significantly improved by the 0.7 factor, and in placebo there is actually decrease in the C-peptide levels. So if you combine these two slides, there is an improvement in the HOMA beta and C-peptide levels, whereas a significant reduction in the HOMA IR parameter. In this small study of 14 weeks, they have shown the effect of teneligliptin on the lipid parameters. If you look at the before and after parameters of lipids, the HDL cholesterol quite significantly increased, having significant p-value. The other parameters are also improved, like the total cholesterol, the LDL cholesterol and the triglycerides, but they are not having the significant p-value. But we can say the trend is towards the favorable. The molecule will not have any negative impact on the lipid parameters. The next dynamic property, the glycemic variability. Apart from the routine glycemic parameters of A1C, FPG and PPG, glycemic variability is also being a emerging risk factor for the management or for the occurrence of cardiovascular diseases. In this particular study, the patients were put on insulin for initial 3 days, and after 3 days they were given the teneligliptin. This is the CGM graph. The upper black line represents the patients taking insulin, and as you can see, there is a sudden glycemic excursions occurring after breakfast, lunch and dinner. Within 24 hours of administration of teneligliptin, there is a sudden decrease in these excursions, and these excursions are decreased even after 48 hours and 3 days after of administration of teneligliptin. In short, teneligliptin will reduce the glycemic variability over and above to the insulin. Moving on to the clinical trials of teneligliptin, which are majorly divided into monotherapy in combination with other oral anti-diabetic drugs, insulin, and the special population of renal and hepatic failure. Before moving on to the clinical trials, I will just like to highlight the current approval status of teneligliptin. This teneligliptin is a Japanese molecule developed by the Mitsubishi Tanabe, which has a very good track record of developing a certain blockbuster drugs. For example, the canagliflozin, which is a SGLT2 inhibitor, which they outlicensed to the Janssen for the worldwide marketing. The other molecules are the telapravir, Remicade and fingolimod, which are very much used worldwide. So in short, the company has a good reputation of developing a research molecule or innovative products. The first approval for teneligliptin came in 2012 in Japan. Korea approved this drug in 2014. India became the third country to approve the teneligliptin in May 2015. Currently, molecule is in phase 2A trials in Europe and US countries. Almost 2.5 lakh patients received teneligliptin during its post-marketing surveillance in Japan. So this 2.5 lakh patient gives quite a good confidence for us to use this molecule in Indian patients. Moving further, the trials are published in well-reputed journals. These are the few monotherapy trials. I will be discussing 1 or 2 trials. And before discussing the monotherapy trials, I would like to just show you the combination therapy trials. As you can see, there are almost 6 trials here. Teneligliptin is studied in combination with metformin in first trial. In next 2 trials, it has been studied in combination with glimepiride and insulin. In this 4th trial, it has been studied in combination again with the glimepiride, but as an open label. In 5th trial, it has been studied in combination with pioglitazone. And lastly, there was one large trial with almost 700 patients where teneligliptin has studied in combination with bigonides, sulfonylureas, glinides like ratiglide, nataglide, and even in combination with alpha glucosidase inhibitors. We will see the direct efficacy or the results of these all trials. As a monotherapy, teneligliptin, when studied for the confirmatory study, the HbA1c reduced by more than 0.6%, the FPG reduced by almost 20 mg per deciliter, The HbA1c target of less than 7% was achieved by almost 41% of the patients, and the postprandial glucose was reduced by almost 48 mg per deciliter with 20 mg of teneligliptin after 12 weeks of therapy. The further results when it has been combined with the metformin. This metformin combination trial was actually conducted in Korean population. The inclusion criteria was type 2 diabetes patients uncontrolled on metformin. If you look at the HbA1c reduction, it is by almost 0.7%, and the FPG was reduced by almost 15 mg per deciliter. The metformin plus placebo arm didn't show any changes in the both A1C and FPG. In this post hoc analysis of phase 3 clinical trials where teneligliptin has been studied in combination with sulfonylurea, glinides, alpha glucosidase inhibitor, and also as a monotherapy, this is the HbA1c reduction results, and this graph gives a fair bit of idea how teneligliptin will perform when used as a monotherapy and as well as very commonly used other oral antidiabetic drugs like sulfonylureas, metformin, glinides like ratiglunide, and alpha glucosidase inhibitors. The A1C reduction started effectively within 2 to 4 weeks and reached maximum up to 12 weeks, and it remained consistent for entire 52 weeks. The maximum reduction was seen when teneligliptin was used in combination with sulfonylurea. The minimum reduction was with the teneligliptin monotherapy. Teneligliptin, when launched first in Japan in 2012, immediately Glenmark approached DCGI for the approval. As per the newer regulation, DCGI asked Glenmark to conduct the phase 3 clinical trial. Along with Glenmark, later on many other companies also approached. DCGI asked them also to conduct the phase 3 clinical trial, but nobody went ahead for conducting the phase 3 clinical trial. Only Glenmark went ahead and conducted the phase 3 clinical trial, which was a randomized double-blind placebo-controlled trial, including 237 subjects across 36 centers, entire India. That means 10 subjects per center. That means patient population was very much heterogeneous. This entire design was as per the suggestions from the subject expert committee of the DCGI. The results, the HbA1c reduction was by almost 0.55% with teneligliptin 20 mg monotherapy for 16 weeks. The FPG reduced by almost 26 mg per deciliter. If you look at the percentage of patients achieving the predefined target of HbA1c less than 7%, almost 43% patients achieved this target, which is quite a good number if you look at the monotherapy design of the trial. And we also look for the effect of teneligliptin on weight, and like other gliptins, it is found to also have weight neutral activity or weight neutral property. Moving further, the special population trial for any new drug when approved anywhere in the world, as per the US FDA and European guidelines, this drug has to be studied in special population of renal failure and hepatic failure to see the kinetic properties and also at the same time the safety. For teneligliptin also, the Mitsubishi conducted the special population trial in Europe. Before moving on to the trials, I would like to just show you the guidelines or the recommendations by these international organizations, particularly for the renal failure and hepatic failure. What they say, if we use a certain new molecules in renal failure or hepatic failure, the kinetic property or the concentration of the drug should not cross the 200% mark or should not be more than 2 times when compared to the normal population. So 200% boundary is the criteria which we need to consider while deciding whether we need to modify the dose of the molecule or not. The special population trial of teneligliptin done in the renal failure patient and hepatic failure patients are published in well-reputed Journal of Clinical Pharmacology and Drug Development. And these both trials are done by the one common author, the Dr. Atif Halabi. We will not go into details of the design and the number of patients. We will just go on to the conclusion, what this trial is saying, particularly the renal population or the renal failure patients. The results indicate that the dose adjustment may not be needed when teneligliptin is prescribed in any form of renal failure, mild, moderate, severe, or even in the end-stage renal disease patient also. The next special population trial of hepatic failure, here also what the conclusion is saying, yes, there will be a little bit rise in the drug concentration, but that rise in drug concentration not crossing the boundary set by the US FDA for the 200%, and this drug can be used safely without dose modification in mild to moderate hepatic impairment. For severe hepatic impairment, we don't have experience, so better to avoid. So in conclusion, yes, in renal and hepatic impairment, there will be a minor effect on teneligliptin kinetic. There will be a little bit rise in the concentration, but that rise is less than the twofold compared when used in the subjects with normal renal or hepatic function. So we need not to modify dose in any form of renal failure and in mild to moderate hepatic impairment. Recently, one pilot study was done on teneligliptin where they included type 2 diabetes patient having asymptomatic diastolic heart failure. The results or the design was 29 subjects with type 2 diabetes followed for 3 months, and they were treated with the teneligliptin 20 mg, and they estimated different glycemic parameters like A1C and D, 1,5-anhydroglucitol, and two important cardiovascular parameters, the systolic function by estimating the ejection fraction and diastolic function by estimating the capital E to small e ratio, that is called as peak early diastolic velocity to basal septal diastolic velocity. At the same time, they also estimated the RH-PAT index, which is nothing but the reactive hyperemia peripheral tonometry for estimating the endothelial function, and the adiponectin levels, which is supposed to have some protective effects against the cardiovascular diseases. The results after 3 months of therapy with teneligliptin, the ejection fraction quite significantly improved from 62 to 64.5 after 3 months, having significant p-value, and the cutoff for the diagnosis of diastolic dysfunction is 12, and by adding teneligliptin it brought down below to the 11.9. So teneligliptin quite significantly improving the ejection fraction and reducing the diastolic dysfunction. The next parameters, the RH-PAT index, which is a marker for the endothelial function, quite significantly improved, and at the same time the adiponectin levels are quite significantly increased with teneligliptin 20 mg. So we can say conclusively that in this small short 3 months of therapy also teneligliptin improves the both systolic and diastolic dysfunction. At the same time, it improves endothelial function and increases the adiponectin levels. So teneligliptin exerts, or we can say it may exert a cardioprotective benefits in type 2 diabetes patients with asymptomatic diastolic dysfunction. If you summarize the total overall safety and tolerability of teneligliptin, approximately 3,000 subjects exposed to the teneligliptin during clinical trial development, out of which 10% experienced the adverse event. Hypoglycemia was found to be the most common adverse event, followed by GI-related adverse events like constipation. The adverse event profile is not significantly different than the placebo. The caution should be there in certain conditions, like when we use teneligliptin in addition to the sulfonylureas or insulin for the risk of hypoglycemia, which is quite true for any other drug also. The another precaution for the intestinal obstruction. If you look at the action of GLP-1, it decreases the GI-related motility, particularly the intestinal motility. So we need to have some precaution while prescribing teneligliptin, or for that matter any gliptin, in those patients who are having previous history of abdominal surgery, to avoid the chances of intestinal obstruction. So it's a class effect, it's not particularly for the teneligliptin. This is another precaution is a QT prolongation. QT prolongation studies are mandatory for any new anti-diabetic drugs, and it has been done for teneligliptin also. And the dose used for the QT prolongation study was 160 mg, which is almost 8 times of the normal routine dosing. And the prolongation was occurred for 9.3 millisecond. If you compare this 9.3 millisecond prolongation with teneligliptin with other gliptins like sitagliptin with 800 mg, which is 8 times of the normal dose of 100 mg, the prolongation was for the 8.1 millisecond. So with sitagliptin it was 8.1 millisecond, and with teneligliptin it was 9.3 milliseconds. So they are quite comparable. So but still we need to have some caution while prescribing the teneligliptin in combination with certain antiarrhythmic drugs like quinidine, procanamide, sotalol, which has a tendency to cause QT prolongation, or those patients who are having the previous history of torticollis pointus. And I think this precaution should be for any other gliptins also. With this total overall safety and tolerability, we will move on to the comparison amongst all gliptins. First of all, the excretion. If you look at the recommendation for dose adjustment for the first 4 gliptins, sitagliptin, vildagliptin, saxagliptin and alogliptin, we need to modify dose when used in the moderate to severe renal impairment. For linagliptin and teneligliptin, as you can see the plus sign, which indicates that they need not to modify dose in any form of renal failure. For hepatic impairment, particularly if you take out the vildagliptin and saxagliptin, other 4 gliptins can be safely prescribed without dose modification in mild to moderate hepatic impairment. For severe hepatic impairment, better to avoid giving any gliptin. Now, if you look at the efficacy of all the gliptins when used as a monotherapy, because we don't have the head-to-head trials, but we can compare the efficacy by looking at the data which is present in the respective PIs, and we arrange this data for all the gliptins, particularly for the 3 parameters A1C, FPG and PPG. First column is for the Saxa, then Sita, Vilda, Lina, Allo, and lastly the teneligliptin. And there is also mention of the number of subjects and dose and the duration of the trial. A1C, the baseline in all the studies was around 8%, and the change from the placebo, if you look at teneligliptin, gives almost 0.9% reduction, which is maximum if you compare all these studies. And also the percentage of patients achieving the predefined target of less than 7% HbA1c. It is also more with the teneligliptin, 40.5, even when the target was 6.8%. So I can say a little bit better response, but I will say that the efficacy is at par with the other gliptins or as good as other gliptins. Similarly, if you go into details of FPG and PPG results for all the gliptins, they are quite comparable. Similarly, we have exercised the efficacy data of all the gliptins when combined with the metformin. Here also the A1C was around 8.5%, and the difference from placebo, the maximum was with the Vilda, almost 1.1%, and with Teneli it was 0.9%. So here also I can say the molecule is performing as good as other gliptins when combined with metformin. The current studies in India, we will see what are the current scenarios or current studies which are planned or going on in teneligliptin. First one is the effect of fixed dose combination of teneligliptin with metformin in Indian population. This trial is registered in CTRA website, which is common platform for the Indian studies. And this trial, the sponsor is Glenmark, and results are expected in coming few months. Apart from this trial, no other trial is going on currently, and that means no other companies are investing anything apart from Glenmark. And as we got the first approval from DCGI for this molecule, as per the regulatory requirement, DCGI asked Glenmark to conduct the phase 4 post-marketing trial to look at the safety of teneligliptin. So for that, Glenmark is about to submit the protocol to the DCGI, and there we are targeting 500 subjects. In this metformin combination trial, which I just described, the target population is 376, and the safety phase 4 trial, the target population is 500. So whatever the studies going on in India for teneligliptin, they all are sponsored by Glenmark. Other companies, they are coming with their own teneligliptin. For that, they need not to conduct any clinical trial. They are coming by doing just bioavailability and bioequivalence study, which is very much simple compared to the clinical trial. And the responsibility of generating more and more data lies on the shoulder of Glenmark. So I will conclude myself saying teneligliptin is a specific DP4 inhibitor with dual excretion through both hepatic and renal route. Because of its long half-life, it's a real once-a-day molecule, and food will not have any major impact on the absorption of the drug. If you look at the efficacy-wise, it decreases or improves all the 3 basic parameters of glucose control, A1C, FPG and PPG. At the same time, when used in addition to the insulin also, it decreases glycemic variability. In one of the pilot study, teneligliptin treatment exerts cardioprotective effects in type 2 diabetes patients with left ventricular dysfunction at an early stage. Like other gliptins, teneligliptin found to be weight neutral and having low risk of hypoglycemia, and overall it is safe and well tolerated. Thank you very much.